Welcome to BioinfoBytes. Okay, imagine trying to find a microscopic needle hidden inside a planetary-sized haystack. Pretty much impossible, right? Now imagine a biological machine that automatically copies that needle over and over until you have a haystack made entirely of needles. That is the sheer magic of the polymerase chain reaction, or PCR, and its advanced successor, qPCR. Let's decode the molecular machinery that reveals these invisible traces of DNA. So let's anchor ourselves to a totally staggering number, 1 billion. When scientists are looking for a single strand of viral DNA, or say forensic evidence, it's completely undetectable on its own. But through continuous, targeted replication, that solitary, lonely strand undergoes massive exponential amplification. We're talking just 30 cycles to turn a single DNA molecule into a billion copies. It's basically a biological photocopier operating in hyperdrive, turning what was once completely invisible into an easily measurable, massive quantity of DNA in just a few hours. To get there, here's our roadmap for this explainer. We'll hit the power of amplification, the 3 core steps of PCR, how we track it all in real time with qPCR, the difference between cybergreen and TaqMan probes, what the quantification cycle actually means, and finally, some real-world applications. Kicking things off with the power of amplification, going from 1 to a billion. If you map this out, DNA amplification grows in a geometric exponential curve. It starts flat, but then it absolutely skyrockets. 2 strands become 4, 4 become 8, and by the time you hit cycle 30, your target DNA shoots up in a steep log phase curve, building a clearly readable quantity right before our eyes. Now, let's break down the actual mechanics of this biological photocopier, the 3 steps of PCR. The core mechanism actually all comes down to extreme temperature control. First up is denaturation at 95 degrees Celsius, which literally melts the double-stranded DNA apart. Next, annealing drops the heat to somewhere between 50 and 65 degrees, so custom primers can attach to our specific target. And finally, extension warms back up to 72 degrees, synthesizing and building the new complementary DNA strand. Now, this entire continuous thermal motion relies on an absolute superstar enzyme called Taq polymerase. It was actually discovered in thermophilic bacteria that literally live in boiling hot springs, so it's naturally heat resistant. You drop it in once, and it powers relentlessly through all 30 thermal cycles without being destroyed by that intense 95-degree melting step. Talk about a resilient little machine. Moving on to real-time tracking with qPR, because sometimes you just need to see the invisible as it's happening. You see, traditional PCR kind of acts like a black box. You wait hours just to see a final product on a gel. qPCR, or quantitative PCR, changes the game completely. It uses specialized fluorescence to detect the exact accumulation of DNA in real time, right during that optimal exponential phase we talked about. As this reaction runs, the fluorescent signal increases directly proportionally to the DNA product. Basically, as more double-stranded amplicons are produced, the chemical glow gets brighter and brighter. Optical sensors map out these cinematic fluorescence curves, giving us precise quantitative data instead of relying on endpoint guesswork. But how do we get that glow? Let's look at SYBR green versus TaqMan probes, which is essentially about choosing the right fluorescent ink. First, we have SYBR green. It's brilliant, but fairly basic. It floats freely until the polymerase builds a new DNA product, and then it snaps into the double-stranded DNA, fluorescing heavily in a vivid electric green. It's incredibly effective, but there's a catch. It will bind to literally any double-stranded DNA, even if it copied the wrong sequence. That's where TaqMan probes come in, because they are far more specific. They sit right in the middle of your target sequence, containing both a reporter dye and a quencher that suppresses its light. When the Taq polymerase physically collides with this probe during extension, it chops it apart. The reporter is suddenly freed, releasing a brilliant amber burst of fluorescence. Crucially, it only illuminates if that exact target sequence is cleaved. All right, so we've got our data, which brings us to the quantification cycle, or CQ, where we measure our starting point. You might have heard this referred to as the CT value in some of the older literature, but the officially standardized term is the quantification cycle, or CQ. Basically, this is the specific thermal cycle number where the chemical glow finally breaks through the background noise baseline and hits that threshold log phase. And here is the inverse relationship that forms the entire backbone of qPCR. A lower CQ value means you had more starting DNA template. If the signal crosses that threshold early, say at cycle 15, you started with a massive amount of target DNA. But a high CQ value means you started with almost nothing, so it required a lot more cycles to reach a detectable level. Finally, let's step outside the lab and look at some real-world applications. By leveraging these tiny molecular machines, we can do some truly amazing things. We can perform vital viral quantitation, like COVID RT-PCR testing, analyze gene expression, and even handle GMO detection. Environmental scientists rely on it to test water for dangerous parasites like Giardia. The food industry uses it to screen for salmonella. And forensic teams amplify vanishingly small traces of DNA to identify individuals with absolute precision. It really makes you wonder, if we can turn a single invisible molecule into a billion readable copies in just a few hours, what mysteries of human health remain hidden simply because we haven't designed the right primer yet? It's staggering to think about how we've transformed biological copying into an automated quantitative marvel. As we keep pushing the boundaries of genomic diagnostics, who knows what invisible worlds we'll be able to amplify next? Thanks for exploring this genetic machinery with me on this explainer. Denature, anneal, extend. See you next time.